In this video, we're going to talk about adaptive immunity, and we're specifically going to talk about the humoral response. So some features of the humoral immune response. This response is going to be antibody mediated. This response is going to respond to extracellular antigens or pathogens. This means pathogens that are not infecting inside of a human cell, things that are floating around outside of the cell, extracellular. The humoral response mainly involves B cells, because remember, B cells are going to differentiate into plasma cells, and plasma cells are going to be the ones to produce antibodies. You're also going to see that helper T cells are usually going to be involved in the humoral immune response. Alright, so let's talk about what this response looks like. So here we have some pathogens, maybe some spirochetes, some bacteria. Alright, so some pathogens get into the body. Okay, so what's going to happen here is there are trillions of different types of B cells that are floating around in your body, but each one is only going to recognize a specific antigen. All right, so this B cell is going to bind to the pathogen, to the antigen. All right, and the place it's going to bind is at this little site. This is a B cell receptor. So in the first step, the B cell receptor is going to bind to the antigen. Once the B cell binds to the antigen, it's going to take that antigen in to its cell. So it takes the antigen in, and it's going to degrade it and chop it up into little fragments. Once these little fragments are produced, the B cell is going to display those fragments on another surface protein, which is called an MHC class 2. And B cells have MHC2 all around the surface of their cell. So the MHC2 is going to display this antigen on the B cell surface. So antigen is displayed. And really quick, MHC2 are specifically displaying any type of antigen that the B cell has taken in. So this antigen may be something bad or maybe something harmless. It depends on what it's ingested. Now, one of two things are going to happen at this step. Now let's pretend that this B cell, the one that we've been working with, is displaying a pathogenic antigen, something bad on its MHC2. So we'll pretend these are fragments of a pathogen. And then let's pretend this second B cell, this other one that's been floating around, and here's its MHC2. Let's pretend this one is displaying something harmless, not a pathogen, a harmless antigen. Now is where the helper T cells are going to come into play. Here's a helper T cell. This helper T cell is going to recognize that this B cell is displaying a pathogenic antigen, and it's going to deliver cytokines. So cytokines are released, and these cytokines are going to activate the B cell. So they're going to activate the B cell. When the B cell becomes activated, it's going to start multiplying. So several of this B cell are going to be made. They're all clones of one another. All right, so trillions of these new B cells are going to be made. And as the B cells are produced, some of them are going to differentiate into plasma cells. All right, so there's a bunch of new plasma cells being made. And remember, plasma cells are going to secrete antibodies. And these antibodies are going to go off and they're going to fight the pathogen. And you can refer to the antibody video that I also made. Okay, and then some of the B cells are going to form memory B cells. So memory B cells are also formed, and these ones will remember this specific pathogen in case the body's infected again with it. Okay, so in this first case, again, the helper T cell activates the B cell so that it produces antibodies, and these antibodies are going to recognize that specific antigen that was displayed here on the MHC2 and destroy it. Let's look at the second scenario. In the second scenario here, I drew another B cell. Again, this is a B cell. But this one is displaying a harmless antigen on its MHC2. This time, another helper T cell is going to come in, but it's going to recognize that this antigen is harmless. We don't want to activate the B cell this time because we don't want the B cell to produce antibodies against something harmless. So this time, the helper T cell is again going to secrete cytokines, but these cytokines are not going to activate the B cell, they're going to inactivate the B cell. And they're going to permanently inactivate this B cell. This is called making the B cell anergic, meaning basically making sure it never activates, or essentially turns it off permanently. And this is one way the adaptive immune system ensures that your body doesn't overreact to something harmless or to part of your own self. Okay? So this is basically the humoral immunity. In both of these cases, a helper T cell was required to either activate or inactivate the B cell.